Hello, everyone. It's my pleasure to be here today speaking about a topic that I believe is critical for understanding one of the most common insults we get in the microbiome area. Those are toxins. So this is going to be directly analyzing different types of these pollutants that we can get on a daily basis that are affecting the balance of the microbiome and therefore affecting the balance of our health. So we do have a very direct connection, and we are lucky that we're getting more and more information about this topic, and therefore we understand much better what's going on in these two worlds and how they're interconnected. And the best part, what we can do for fixing what's going on in terms of hypertoxicity and microbiome imbalance. I always say that the microbiome can only be as healthy as the terrain that these microbes are living in. If you don't have a healthy terrain, if you don't have a healthy ground for these microbes to live, it's very unlikely you're going to get a balanced composition in your microbiome. So even though probiotics and prebiotics and other strategies, they can be helpful to a certain extent, and they can help regain a certain balance in the microbiome field, if you don't clean up that environment, if you don't fix those conditions, you're never going to get long-lasting results. So we have plenty of evidence of how the microbiome is so involved in human health, and we know that any imbalance can lead to very, very serious chronic illnesses, for example, Alzheimer's or metabolic diseases, Parkinson, autism. We have so many things that are connected directly to the healthy status of the microbiome, but also we know that toxins are critically related to multiple of these chronic metabolic conditions. So we see an overlap where when we analyze the toxins, we see that they're literally connected directly to the occurrence and the severity of these type of chronic illnesses. And also if we look at the microbiome field, we see the same type of connection. So that, of course, got my attention over the years, and I start seeing the overlap. And then the question was, well, are these two variables connected? Are these two different type of modulated agents connected somehow, that they're actually contributing to build up and the risk factors for creating all these chronic illnesses? And the answer, of course, I'm going to spoil this, is actually yes. So we're going to talk about what is microbiome toxicity, how heavy metals, environmental toxins, mycotoxins are affecting the gut microbiome, and the toxins also, they are a very, very, very important factor in terms of maintenance of the terrain conditions. And then we're going to look at strategies for rebalancing the gut microbiome, but with the very intentional focus on detoxification. So that's going to be our outline for today. So the microbiome world is actually very complex. It's very intricate. We have multiple types of microorganisms living in communities, and we have different types of complexities in terms of where we are looking at those microbes inside the body or outside the body. So we know that the vast majority of them is located in the gut, and they're contributing for a lot of vital processes like digestion and immunity and helping down to break down complex foods. And they're making also short-chain fatty acids, and they're upregulating our immune system to create a defense line against anything that can be coming out from the outside. Also, they're reading information from the outside to help us understand better what's going on, and of course, respond accordingly. And we know the gut-brain axis is a huge piece of the puzzle, and all the mood, the cognitive functions that we are relying on to be healthy and thriving, they're actually related to the status of the gut microbiome as well. And also the microbiome is related to the production of neurotransmitters for making this happen. 90% of our serotonin, 50% of our dopamine are produced in the gut microbiome. So this is a really, really important aspect of our health. And definitely we need to keep this in a healthy balance. And that's why we're talking about this factor right now, toxins. So we know if you go, this is microbiome 101. If you go to any literature, you're going to see that if we look at the causative agents that are damaging our microbiome, of course, sad diet, processed diet are definitely not good for the microbiome health. Stress, any form, any kind of stress will affect the microbiome. Sedentarism, trauma, of course, it's another huge one. The last ones that I'm mentioning here, which are heavily related to the status of the microbiome, high exposure to toxins, high accumulation of toxins, which are two different things. And the third one, it will be poor excretion of toxins. So when we talk about toxicological issues, it's not just about the toxins we're getting exposed to, it's the relationship we build with those toxins. Of course, the ideal world would be no toxins, but the challenge that is created over the exposure to toxins also has an direct activation of certain pathways in the body. And if we're able to cope with these toxicity issues, we might not get out of balance. There's some organisms in the world that can survive in very high levels of toxins and they do fine. It is all about our pathways, how open they are, how able are they to cope with these type of challenges and stay on a balance point in an equilibrium between what's coming inside to the inside of our body and what we can eliminate. If we have affected pathways in any of these areas, if we're not able to excrete the toxins properly, or we accumulate in these toxins for the same reason, or we have too much exposure, more than what our physiological pathways can handle at any given time, then we will have a problem with these toxins and they will create imbalances in different aspects of our physiology. So when we talk about the definition of gut microbiome toxicity, it's about the functional damage that is created by the exposure of these xenobiotics or external toxins and the endotoxins produced by imbalanced microbiome patterns. So whatever bacterial metabolites we're producing, that is creating affected pathways in terms of what we need to be produced from the microbiome standpoint will directly, of course, affect the whole balance. And that is what is labeled or named as microbiome toxicity at the end of the day. And we know that the impact that we can get from xenobiotics in the gut microbiome can be direct or indirect. So we have some environmental chemicals that can go directly to the gut microbiome, to the gut bacteria, and they will interrupt specific metabolic pathways or gene expression. So they're going to change the microbiome in a certain direction, and that will create a selective process in terms of what survives, what's not surviving, and that will create a different type of beta diversity. Now, also some chemicals can indirectly affect the gut microbiome, and this is critical to understand because it can affect the host physiology, meaning that they will affect the mucus production, or they can affect the communication between cells to cells. They can affect the liver function. They can affect the kidney function. They can affect their immune system. So many things that are related to the terrain balance and therefore maintaining the equilibrium in terms of the microbiome composition. So we can get these two pathways affecting the microbiome in this way. And I think that at this point, we are in a good place. We can say with a lot of consistency that the gut microbiome toxicity, it's another aspect of the overall assessment in terms of toxicity that can be affecting our bodies. So we look at brain toxicity, we look at liver toxicity, we look at kidney toxicity, but we're not looking at directly what is gut microbiome toxicity when we try to analyze the overall toxicity issue that can be affecting one person's health. So I think that aspect is being relegated to a secondary place, and it has to become more of a priority in terms of analyzing people's conditions and what is driving this chronic metabolic illness into their physiology. And that's why we're going to stop and look in detail what's going on in terms of heavy metal presence, environmental toxins, mycotoxins, and endotoxins that could be potentially affecting and damaging the gut microbiome. So first of all, let's start with the heavy metals and the gut microbiome. If we go and look at the timeline in terms of what we know of the gut microbiome pathways and the analysis we have been doing in terms of the exposure of these microbes to different types of heavy metals, we can trace this back to the 70s, and we know that we start doing some research about modifications in animals, especially ruminants, that were exposed to certain types of heavy metals like cadmium, chromium, mercury, arsenic, and lead. And we saw some changes there which were significant, and that gave us some clues. And we started doing more and more research about these type of modifications within exposure to certain types of heavy metals up to today, when we have direct evidence that we can confirm, for example, things like cadmium inducing gut microbiome dysbiosis and aggravating liver injury because it's increasing gut permeability in mice that they have, for example, affected microbiome compositions. So we know that the microbiome actually plays a dual role in coping with the exposure to this type of toxins, and we're going to talk about that more in detail. The mechanism of the antimicrobial activity of heavy metals is mainly by producing damage directly by building reactive oxygen species. And we probably, all of us, we have heard about these ROS being very damaging if we can cope with those on a very consistent basis. Well, of course, we have pathways that we can upregulate NRF2, etc., that can produce antioxidant responses and therefore cope with these reactive oxygen species on demand. But if we surpass the possibilities of the body for coping with the reactive oxygen species challenge, that will create damage to the tissues, but also we see a damage in the microbiome spectrum. And that affects the way that the microbiome can produce enzymes and the protein expression within their cells, and the membranes, the actual anatomical structure of the microbes in the microbiome can get compromised. And therefore, of course, all the nutrient uptake and the communication pathways and genotoxicity. We can damage the genetic information included in those cells because we're damaging the actual anatomical structure that is protecting those genes for getting damaged with all these type of exposures. So the gut microbiome actually can have these direct factors that are influencing the composition and altering the profiles because they're getting damaged by the exposure to this type of toxins, heavy metals. But also, we have the ability of this gut microbiome to actually increase certain populations of microbes or producing certain metabolites to cope with the damage that is created by these heavy metals. And they can, then down the road, they can reduce the absorption of heavy metals in the gut, and they can help converting heavy metals to a less toxic form. Actually, that can be done also in the opposite route and can become more toxic if they go to certain pathways in the gut microbiome, but also they can help reducing toxicity by creating binding and scavenging agents that can eliminate faster the heavy metals out of the body without getting them reabsorbed. So that will depend a little bit on your healthy baseline of the microbiome that you had by the moment that you get exposed to these type of heavy metals. So, and at the end of the day, maintaining a healthy microbiome will help coping better with heavy metal and any other type of toxin that you can get exposed to. So we know the gut microbiome can affect directly or indirectly, again, can get affected directly or indirectly by heavy metals and any other type of toxin actually. And that will alter the composition of the gut microbiome itself. Now, this could be, again, a coping mechanism. So we see some things like, for example, metabolites produced by the microbiome, like hydrogen sulfide. And we see that as a very bad sign. We think right away that there's something wrong with the microbiome. In many cases, those metabolites are actually produced to be able to eliminate faster and better those heavy metals out of the gut. So in many cases, the pathways of the microbiome are working to eliminate and become more efficient in detoxification of these heavy metals. We see the end products and we associate those things with dysbiosis and pathological conditions, but not all the time are related exactly to just a negative factor. They can become produced in larger amounts to compensate the lack of ability of the body to eliminate these heavy metals out of the system in an efficient way. So that's a little bit of how I will suggest to see the composition of the gut microbiome, but also the metabolites being produced by the microbiome with an aim of more compensation and help in reducing toxicity inside the body. Now, also we know that the microbiome can help excreting heavy metals by multiple enzymatic pathways, and that has been proved by research being done in mice. And when we get an antibiotic-treated or germ-free mice, we see that they are able to excrete much less heavy metals, including cadmium, lead, methyl mercury, and arsenic in their stools. And they start accumulating more concentrations in their blood of these type of heavy metals compared to control mice that they have a healthy populated microbiome. So we know that the microbiome is playing a role in terms of decreasing accumulation and improving elimination of these heavy metals from the gut. Also, we know that there's some microbes, like for example, Pseudomonas, that they can produce something called cytophores. Cytophores are like iron, for example, chelating molecules. They're especially good for chelating these heavy metals out of the body. That includes lead, cadmium, mercury, chromium, arsenic. And they form insoluble complexes that can be excreted more easily, and we prevent reabsorption of them inside the gut. Also, bacteria like Oxalobacter formigens, that is an oxalate-degrading bacteria, and it's a commensal bacteria. Actually, it's a keystone species for the human microbiome. Can actually bind lead ions, and they can transform them into lead oxalate, which will reduce, of course, the possibility for absorbing lead inside the intestines. So it's actually a very important keystone, not only for degrading oxalate-containing foods, but also helps binding out lead out of the body. And these are a few tips that I would like to give in terms of analyzing the microbiome with a different angle, and now see the benefits of certain things that we can sometimes demonize and think these are just signs of dysbiotic microbiomes, and they might be just compensating and even helping us reducing toxicity from an underlying hypertoxicity issue that we have not assessed yet. And also, it can pose the question, is this person making more hydrogen sulfide, for example, or making more of this metabolite just because it's trying to deal with, in a better way, with the hypertoxicity problem that is underlying that we have not seen yet? And that's why I love testing. I know that there's some issues with the testing. Sometimes it's not that accurate, but there's actually absolutely no perfect testing in the world. So I take testing with a huge appreciation because it lets me know how much toxicity load we have in our body. And to a certain extent, but still, it's useful. We can know what is there, what is passing through, and what we can do for improving the accumulation and improving the excretion of these toxins, no matter what. So yeah, this is something I'm highly interested in. And I know there's room for improvement. We need better testing systems. They're going to come over time. I know that because this is becoming a very interesting and also very trending type of issue, and it's becoming more correlated with all the chronic illnesses we're seeing today. So definitely, I think that there's going to be more and more intentional improvement on these areas. Now, the heavy metals also, they can affect the composition of the microbiome in a very important way. We have seen that at the phylum level, there are studies demonstrating that the abundance of Firmicutes and Proteobacteria can decrease, and the abundance of Bacteroidetes can increase after certain heavy metal exposure. And all these heavy metals also, they can create time-dependent changes in the microbiome. And when we test mice, for example, Akkermansia muciniphila, which is such important keystone, it can be significantly decreased after getting exposed to the heavy metals like cadmium, lead, copper, and aluminum. So those are things that we need to be aware that sometimes our gut composition will be changing, not because of our diet alone, it's just because we're getting exposed to toxins. Now, we have a good diagram in terms of what could be potentially affecting the gut microbiome from the heavy metal side. There's many ways that bacteria in our gut can actually create metabolites and increase the abundance of these heavy metals. But also, in some cases, things goes in the wrong direction, and we can actually convert things, heavy metals, to a more toxic form. And that will depend, again, on what your baseline microbiome composition looks like at the time of the exposure. So working on your microbiome in an active way can be one of the most profound ways you can actually prevent creating more hypertoxicity over time. So that's something that I believe needs to be encouraged, not only for your nutritional needs, not only for your immunity needs, which are very important. This is also another factor that allows me to understand even more why it's so important to have a microbiome that is balanced. So these changes are highly related to the balance and the metabolic pathways of the microbiome of the gut directly. Now, if we see this diagram I'm looking at here, we have all these different types of toxins coming from the outside, xenobiotics, antibiotics, heavy metal, pesticides, even artificial sweeteners are listed here. And this is a little bit of the explanation of what I was trying to give before. Those are going to create functional changes in your body's pathways and in the microbiome. And then the microbiome will either change diversity-wise, it will change the way that it's producing metabolites, and that can create multiple types of chronic illnesses because there's going to be a change in the way that we make things inside the gut that are definitely related to our overall health status. Now, what about environmental toxins? Environmental toxins are a huge deal because environmental toxins are all over the place. Right now, we know that we have a tremendous amount of PFASs and plastics all found in the water supply. And that along with the metals that we see, of course, as well in the soil, in water. And so everything is getting added to this pool of toxicity that we're exposed to potentially on a daily basis. Now, we know that humans are exposed to hundreds of chemicals and more are being released every year without knowing what will be the direct effect in our physiology. And some chemicals, they can live a long time in our body and in the environment. And there PFASs, for example, those are called forever chemicals because we don't know how long they can actually stay in the body. There's no timeline that we know that will tell us they will stay a certain amount of years. We know that they can stay for our understanding, they can stay forever in our bodies. So that's a complicated scenario if we think about that one exposure can live forever in our body. And we don't get just one. We get an ongoing exposure most of the times of all these chemicals. And they will create gut microbiome issues and toxicity. And we know there's a lot of documented impacts in terms of the microbiome balance. So we're going to talk about those things. So, and again, we have this multiple way that we can get the microbiome affected. In some cases, we can have the environmental contaminants directly affecting the bacteria composition. And in other ways, we can actually affect the host metabolic activity, changing the patterns in the terrain and the way we modulate the conditions for our microbiome to be healthy. So first one, we're going to talk about BPAs. Well, BPAs are actually highly used still. I know that we see now plastic bottles, and we're going to talk about that they say BPA free, but just that's one of that's the tip of the iceberg. We have so many other types of BPAs used in clothing and in food processing and millions of other pathways that are highly relevant and in terms of the exposure that is created. And that's why we need to be very careful about what we're using on a daily basis to try to reduce the exposure as much as possible. But we have limitations for this. And unfortunately, we have a very potent damage by the exposure of the body to this type of pollutants. And even though the BPA life is inside the body is very short, it doesn't really stay in the body for too long. Just that short-term exposure can create a lot of damage in the human cells, but also for the microbiome. The BPA can alter the gut microbiome in a variety of ways and in a sex-specific manner. We know that there's differences in terms of the damage created by the exposure to BPAs in that is different in male and females. Also, we know that there are some upregulated levels of certain bacteria, in this case, for example, Methanobrevibacter, which they will, and this has been seen, especially in males, and that can create different type of relationship with exogenous fuels, and that can upregulate the host energy intake and ultimately relate to weight gain. So there's a lot of things that we can see in the toxicity spectrum that can affect the body weight that are not diet related, are toxin related. Also, we know that there are some induced microbiome changes that can increase the risk of carcinogenesis. So this is not good news when it's elevated inside the body. It is definitely a trigger of inflammation, and it can affect the production of certain fatty acids. It can also create chronic intestinal inflammation and also hepatic inflammation and lead to metabolic disorders. Also, there's a certain decrease in the relative abundance of Firmicutes and proteobacteria. So we're going to have pathways in terms of the production of certain metabolites affected by the decrease of these type of films. So definitely everything goes a little bit upside down. And again, this is it has a very short life inside our body, but it is the ongoing exposure that creates the most considerable amount of damage inside the body. So that's the main problem. We have too much of this incorporated in many commonly used products and clothing, etc., foods, and even soil. And there's not too many ways to avoid them. And when we look at the BPA alternatives, we don't have that much information compared to the BPA. But when we look at the BPA alternatives, we can see in the initial studies that they can be same or even worse in terms of the damage that can create to the microbiome. There's development issues and multiple other forms of changes in the microbial communities based on the exposure to all these alternative plastics that are not BPA based. Also, we have some other environmental toxins called POPs or persistent organic pollutants. That's what it states for. And we have things like, for example, polychlorinated biphenyls or pesticides like DDT that are in this category. And we know these things, they stay for a long time in the environment. DDT is banned, and still it's being detected in sample of people that they didn't even were born when DDT was allowed. So they're just getting a secondhand exposure by the soil and the produce that is getting contaminated by the accumulation of this pesticide by the time that it was used. So it's still there, even though we don't use it anymore, in farming at least. And perchlorinated biphenyls, that can actually increase, those can increase gut permeability. They can upregulate the expression of interleukin-6 in the gut. And so definitely an inflammatory marker and in the gut and especially in the development of mice, not even in full-grown mice. We're talking about mice that were exposed through the maternal diet. So this is as early as it can get. So it's a considerable factor because not only we're concerned about the subject itself, is what it's from. It can be from the maternal feeding that you can get this type of toxins incorporated in your body, and of course this can affect the whole development of the newborn later on. So it is not a small issue because again these POPs are very, very long lasting in our physiology and in the environment, and they will create long lasting damage. Now, when we talk about pesticides, like we're talking about DDT, we have now other forms, other versions, and unfortunately they're not better. The most probably famous one is glyphosate. And glyphosate can disrupt the microbiome in so many ways. First, it affects something that is called the Shikimate pathway, which is necessary for the production of essential amino acids in bacteria. And that's how they get away with this, because it doesn't really affect any cellular pathway inside our human physiology, but it does affect the ability for producing essential amino acids inside the cells of the bacteria of our microbiome. And when that happens, this is the downside, not only creates direct damage to the microbiome, but also there's some bacteria that can actually handle this. They don't really rely on the Shikimate pathway. And those are actually the ones that we like the least to be increased in the microbiome composition. So not only is killing microbes directly, it's also imbalancing the microbiome composition by this type of Shikimate pathway resistant, let's call it that way, type of bacteria. So it creates more an imbalance than anything else. We know also that it affects directly our metabolic functions, so it's disrupting processes that are necessary for our health, including, of course, the production of short-chain fatty acids. It will affect the barrier. It creates leaky gut quite fast, actually, and the immune response, of course, gets altered, and that on a chronic basis can affect many, many other things in our physiology. Diversity gets affected, of course, because we're killing microbes every time we get exposed to, and this is a water-soluble molecule, so it can go everywhere. That's the worst part. It doesn't get controlled by the liposoluble mechanisms we have for binding and controlling toxins inside our body. This is water-soluble. It's way harder to get controlled, and therefore, it's easier for this molecule to go everywhere inside the body. And we know that that can create also leaky brain, leaky skin, and so many other aspects that can affect our health. And therefore, it is a very concerning thing that is being used so massively in traditional farming. And this is only one associated with all the other types of toxins we see nowadays, 2,4-D, etc. So we definitely will have an overlapping problem in terms of the amount of usage of all these pesticides nowadays, and the environmental and the human health implications that they pose by getting exposed to these things. Now, mycotoxins. Mycotoxins are naturally occurring in the environment. We have toxins produced by mold every single day. They're out there in the environment, and they don't really make us sick when they're produced outdoors. The problem is that when they start getting produced inside the house, because we have mold growth in the house or in the office or in the school or in the car or anywhere that you can find them, and because we're insulating our buildings to try to become more energy efficient, there's an increased amount of accumulation of them, and that increases exposure. And of course, in many cases, it can overwhelm the detoxification pathways related to them, and it can create illness. And the illness and the affected pathways not only relate to the human cells, they also relate to the microbiome composition. We have multiple types of mycotoxins: aflatoxins, ochratoxin A, trichothionthanes, cerulon, phimosins, to name a few. They're coming from different sources typically, but all of them, they produce a certain effect in the body, and in high levels, they can create a lot of issues in our health. They typically create chronic health issues, and we know they're quite damaging for the liver and the kidneys, and they can lead to chronic inflammation on those organs, and ultimately can derive into cancer and other issues. And neurotoxicity is another reported problem that we can get from the exposure to these toxins. Reproductive development toxicity, it is another issue. We have seen hormonal-like alteration when we get exposed to certain types of mycotoxins. And like I mentioned before, the kidney and liver damage is one of the worst parts. And you have to always keep in mind that kidney and liver are taking care of the terrain conditions of your microbiome. So how potent is that? How important is to keep those organs working in the proper way? Because pH and the degradation of certain nutrients and availability of certain nutrients and peristalsis and detoxification, the activation of certain nucleus in the cells, all of those things are related to a healthy function of these organs, the kidney and liver. And if we don't have that ability, we're going to affect the whole terrain of our microbiome, and therefore our microbiome gets damaged. So yeah, that's a critical pathway that we can get affected by the exposure to from mycotoxins. So we can get direct toxicity. The mycotoxins, by definition, are antimicrobial, and that's why we can see imbalances in the gut microbiome directly coming from the exposure to from these toxins. But also, again, because of the metabolic alterations, we can see damage in the gut microbiome. And also, we know that they have an ability for impairing the gut barrier function and leading to leaky gut. So that's another factor that contributes to damage by the exposure from mycotoxins. And by the way, the immune system function is going to get affected. They're typically immunosuppressive, and that, of course, affects the way we correlate when we check for any type of opportunistic pathogens, etc. And the detoxification pathways. Again, we don't have a good functioning liver, good functioning kidney. We can't excrete toxins properly, and we can produce enough bile. We can conjugate things properly. We won't be able to eliminate things on the proximal tubules on the kidney, and that directly affects the amount of accumulation of toxins inside our body, and that affects the whole microbiome composition as well. So definitely a big deal. Now, the last but not least that I would like to mention today are endotoxins. Endotoxins are made by gram-negative bacteria typically. They are present in the cellular wall of the gram-negative bacteria. They're embedded in this membrane. When these microbes die or they get killed, whatever happens first, they will release these lipopolysaccharides or LPS into the environment. Of course, the larger the populations of gram-negatives that we have in the gut, larger the release of these lipopolysaccharides. Now, in the normal circumstances, these are actually contained inside the gut. They can create a certain challenge to our immune system, and they can actually play a healthy role in our microbiome physiology. Also, we do have a normal dominance of gram-negative bacteria inside the microbiome composition that is healthy. We expect to have that. But beyond that point is when we get these elevated numbers of lipopolysaccharides, or sometimes we have too much gram-negatives in one specific area of our digestive tract, and that leads to hypertoxicity caused by endotoxemia. And at the end of the day, there's a term that we have that is called metabolic endotoxemia that is associated with metabolic disorders related to an increased amount of release of lipopolysaccharides. So we know that all these metabolic disorders, including type 2 diabetes, obesity, and cardiovascular disease, they're highly related to the expression and the maintenance of these lipoproteins inside the body. And that is huge, because this could be the one single root cause that can explain in many ways all these chronic illnesses. So we have a model that explains the circulation of these toxins, and we can see that the lipopolysaccharide producing bacteria, they can go into circulation, especially when we have a leaky gut issue. So they can cross the barriers through the disrupted tight junctions, and they get into systemic circulation. And then, of course, they get circulating all around our body, and they can disrupt also the function of the liver, and that can increase also the expression of certain immune cells that ultimately will expose those molecules to immune cells like monocytes, macrophages, neutrophils, and those are going to increase the inflammatory response, and that creates what we call meta-inflammation. So it goes everywhere. They start traveling around the body, and that will affect the function of multiple areas. And actually, every single place where blood goes, we can get inflammation through this endotoxemia pathway. So it can relate to pretty much anything we know in terms of chronic illness. Now, the other problem is that when they affect the liver function, that can contribute to something called cholestasis. And cholestasis is exactly what we don't want in terms of detoxification, because that will bring all the circulation of conjugated toxins back into circulation. So the normal pathway will direct things from the blood circulation through the different phases of detox until they get excreted in the bile canalicles. But if that doesn't go in the right direction, this actually reversed, and we start pushing toxins inside the blood again. And that, of course, make them recirculate through the body, creating, of course, all the damage we know in terms of inflammation and tissue damage by the expression of ROS. So we want things to go out and not come back in. And that's why it's so critical to keep this lipopolysaccharide release contained inside the gut space and eliminated properly. And for that to happen, you need to have, of course, a balanced microbiome. In absence of that, you need to have proper mechanisms to cope with that until you regain the balance. And we're going to talk about those things. We have tools for helping through the process while we're getting a better balance in the microbiome pathways. So all this is related to an immune activation disorder. So ultimately, what we want is to keep inflammation in a certain balance. And for that purpose, endotoxemia needs to be controlled. And if not, this again leads to so many illnesses like heart disease, liver problems, hypertension, type 2 diabetes, dementia, cancer, reproductive issues, and non-alcoholic fatty liver disease, which is critical for, of course, our health. So these are things that we need to consider. So what we can do about it? That's the main question. I think we proved the point that toxins are highly damaging for the microbiome. They can drive changes in the microbiome composition that, of course, they will create imbalances in the production of metabolites. They will create imbalances in the patterns on the composition of the microbiome, etc. So we know those things are not good for us. Now, we need solutions. Well, one thing I mentioned before is testing. I like to know what we have inside. And even though, again, there might be some flaws in the testing, and we can test different samples. We can use an urine analysis, we can use a hair analysis, or we can use blood analysis. We can use all of them together. And there's different price ranges for testing for those things, or they could be other factors associated. At the end of the day, I want to have a baseline. The most important reason why I want to have a baseline is not because this is going to give me a super accurate vision of what's going on. It's going to give me a certain degree of a baseline. Again, arguably could be more or less specific, But what I want to know is when I start a protocol for detoxification, where I am in terms of toxicological burden and when to finish. If I don't have established baseline, I don't know when to end. I don't know if I'm making progress or not. I don't know how to follow until I get to the result I want. And that's why for me it's critical to have analysis for that purpose. And I use urine samples for most typically. I don't like to provocate. That's a side note. We can discuss that. But provocation can be useful in certain cases, but also some people can crash if you use DMSA, which is one of the most typical agents for provocation. So I like to see what the body can eliminate on a natural way, because that's how I'm going to work at the end of the day. I'm not going to add a very strong provocative agent on a frequent basis. I'm going to use upregulation of the excretion pathways to make things work better for my body to eliminate toxins better. That's what I want. And avoid reabsorption, which is critical. So for me, the rebalance strategies for the microbiome to get better through detoxification are reducing the toxicological load, avoiding exposure. That's a huge one. That's one of the most profound ways you can detoxify, is eliminating the source of toxins. And that could be either foods, or it could be your environment, your house, your car AC. It could be multiple. I think we need some time for dedicating ourselves to understand where this could be potentially coming from. Improve your terrain conditions. And this is all about your support organs. Support your microbiome through the process, after the process, and probably forever. Not probably, actually, forever. And improve your lifestyle and diet, because that's also going to give you a profound change in terms of what you can do for eliminating toxins more properly and also avoiding potential sources of toxins. Probiotics are useful, and we have some strains that they have been proved to be very beneficial in terms of reducing toxicity in the gut. We have certain strains of Lactobacillus plantarum, which they have been used for chromium toxicity, cadmium toxicity, lead toxicity, and there's a strain of Lactobacillus brevis, which is also used against mercury toxicity. Some of them can be found in certain commercial products. There's a company called Microbiome Labs. They have one product that is called Megametallic, that they have one of these strains of Lactobacillus plantarum that helps tremendously to bind these toxins out of the body. This is a ghost probiotic. It's a dead form of a probiotic that has a very specific exopolysaccharide in the wall that can bind these toxins out of the body. And that is really a selective process that is very benign, doesn't create any side effects. I think it's a fantastic add-on for detoxification strategies. Now we have a good amount of research about this strain, so I think that we can try to find them. The one I know, again, the most is Megametallic. It could be other ones out there, but that's the one I've been using the most for my detoxification protocols. Also, probiotics can stimulate peristalsis and can stimulate the function, the support organs, which is also critical for maintaining the flow of mobilization of all these toxins out of your body. If you're constipated, there's absolutely no way you can detoxify properly. If you're dehydrated, you can't detoxify properly, by the way. Now, there's some other probiotics that can actually increase the production of certain metabolites. In this case, for example, Phycoalterin peroxide, which is a keystone species, has a gene that encodes a methyltransferase, and that can improve the arsenic detoxification, and also limits the absorption and accumulation of lead in the host tissues, and also it will upregulate the production of short chain fatty acids, and therefore improving the tight junction protein expression, which also reduces the risk of absorption of these toxins inside the enteric circulation. Cremancin muciniphila, we know that improves the gut barrier function, of course, helps reducing as well the recirculation of toxins. So we have different tools that we can use from the microbes, from the probiotics that we can use for detoxification purposes. I like also a formulation that is also from Microbiome Labs, which is Megaspore Biotic, because it upregulates so many pathways that are related to a healthy functioning of the microbiome and the organs, body's pathways. So that helps with the immune function, with our detoxification, with antioxidant production, etc. We need that support through the whole process. The other thing that I use pretty frequently are immunoglobulins, because those are especially good for binding endotoxins. So these are naturally produced in the body, but we can get them concentrated from bovine serum or bovine colostrum. And in that way, we can amplify that effect, and we can not only support the immune function, but also we can produce this binding effect. It's a biological binder to reduce the endotoxemia accumulation. So they play a role in immune function. They also help protecting against pathogens. They help keeping the maintenance of the gut microbiome balance by neutralizing these bacterial-derived toxins. They also help reducing inflammation pretty dramatically, and again, because they will neutralize pathogens that can produce metabolites that can be pro-inflammatory, and that helps a lot for reducing overall inflammation that also can lead to, which affects detoxification, and they help also improving the gut barrier function. So that's critical for everything we spoke before in terms of reducing reabsorption. At the end of the day, we want to keep the flow going, and that our cellular detoxification goes into the blood and then excreted. All these toxins are getting excreted through the kidneys, or they get into the liver and then they get conjugated and excreted into the small intestine. So we want this transportation of toxins and elimination of toxins to happen in a very, very diligent way. Now, when it comes to solutions and detoxification strategies, I know it's tricky. I will mention one system that I've been using that is pretty general and very efficient, that you can be used. It can be used in multiple scenarios. Some people will need more specific support. They will need more specific approaches in terms of dosing on or products. But I think that overall, this will help you to give you an overall support in terms of detoxification. And I've been speaking quite a few times about this, and this is the Advanced Butch-Cash Detox System that it was developed by Quicksilver Scientifics and Dr. Shade. So we have a product called Liver Sauce. We have a binder called Ultra Binder. We have Kidney Care. We have liposomal glutathione, and we have a product called Membrane Mend. So this is actually a very comprehensive package because it puts together products that will help from step 1 to step 2, 3, 4 until the end, the proper elimination of toxins. But also they upregulate functions of support organs, which are needed for maintaining normal balance in the microbiome. So you're working on these organs and you're helping the microbiome to stay in balance because you're improving the terrain conditions, because you're keeping the bile flow. You're reducing toxicity. You're removing things that are not supposed to be in certain areas of the digestive tract. You're balancing the pH. You're increasing the likelihood of creating fermentation because you are also maintaining a certain balance in the fermentative microbes, and that leads to a better anaerobiosis that is needed for growing Akkermansia eosinophila, for example. So all these things are really needed for a healthy microbiome to occur. So I like, because the push phase is done by Liver Sauce and glutathione, this taking care of phase 1 and 2 of detoxification in the liver and in the cells, and upregulates NRF2, which we know has such a profound way of improving antioxidant pathways and also keeping the flow of the bile in the right direction. And then we have the phase 3, which is the Kidney Care and the Ultra Binder, which are related to the proper excretion of toxins from the kidney source, from the kidney pathway, and also binding these toxins in the intestines so they don't go back to circulation, and instead they get eliminated properly out of the body. And this is critical because most of these toxins are able to cross this barrier and go back again into circulation. But we properly bind them to certain agents like activated charcoal or chitosan or zeolite. We can actually create these complex molecules that can cross, and they will be eliminated out much easier. So the Liver Sauce has multiple ingredients that will improve bile flow generation, but also they create more immunotolerance and they stabilize mast cell, which again, we try to do that because we want to use this type of nutraceuticals because we want to keep inflammation downregulated to the point that it will not create cholestasis. And we have these NRF2 upregulators like them and our lipoic acid and Celmarine, which is coming from Melphistil, which also is an amazing support for keeping the bile flow out of the hepatocyte and into the bile duct. So all these things together, they keep the bile flow mobilized. And that's what we want, because we want these toxins that are coming from the blood into the hepatocyte to go through the different phases of detoxification. They will get conjugated by these amino acids, and then they will go out of the hepatocyte and into the bile canal. If we don't keep that process going, we get things going back to circulation, which is exactly what we don't want. So again, very important aspect of this detoxification is keeping the bile flow moving. Probably one of my favorite organs to work in because of the amazing results I get every single time is the liver. Liver is critical organ for human health. And the role of binders is critical. We bind the toxins, we prevent absorption of toxins back into circulation, and we help these toxins to get excreted out of the body. And this ultra binder that I'm mentioning right now has IMD, which is a proprietary blend of silica that is trademarked from Quicksilver Scientifics. We have charcoal, we have zeolite, cytosine, acacia-gum, and aloe. And acacia-gum and aloe are just soothing the gut, so we prevent a little bit of the harsh effect that these binders can create inside the intestinal tract. Also, we have digestive bitters. Digestive bitters are probably one of my most favorite support for improving bile flow, but also for stimulating the digestive function. We don't have too much bitters in our foods nowadays. Unfortunately, that was intentional in terms of removing those bitter tastes because they're not really appreciated by the average consumer. But they're highly 100% needed for stimulating receptors in all our digestive tract, that they have a very direct impact in the functionality of our digestive system and our immune system. So they work just way beyond the digestion. They do help with digestive enzymes and the liver and gallbladder activity and the gut motility, but they also propose anti-inflammatory properties. So I think this is a really, really good idea for using them on an ongoing basis. They're so safe. And there's some cultures, there's some parts of the world that they are being used traditionally, and they're part of pretty much any major meal of the day. And they can help putting this bile out of the hepatocyte, stimulating MRP2 receptors and BSAP receptors. So we have these two ways for pushing out conjugated toxins or directly bile into the bile canicle, and that can be upregulated by the bitters function. That's great. Also, they upregulate NRF2. They have many benefits in terms of bile flow. Another thing that I like to use for upregulating detoxification is nitric oxide. Nitric oxide is a very, very important molecule that we can make endogenously by L-arginine pathways in the endothelium of our blood vessels. But above the age of 40, we lose the ability because of an enzyme that is related, the nitric oxide transferase, synthetase, that is affected and is not being produced properly. And therefore, we rely on nitric oxide conversion by the nitrates we're eating. And unfortunately, our foods right now, doesn't have too much nitrates in concentration. So supplementation can help compensate in the lack of nitrates, that they can be reduced into nitrites and ultimately reduced into nitric oxide. And nitric oxide, it's a key in the red blood cells for exchanging oxygen. So if you don't have nitric oxide, you won't be able to provide oxygen to the tissues. And that is so important for many things, including maintaining normal conditions in your gut terrain and also for detoxification purposes. So besides the blood flow, and we have immune responses that are upregulated by nitric oxide, and we have an oxidative stress modulated by nitric oxide, that will help with the pathways of detoxification and again preventing cholestasis. And we have also the activation of enzymatic activity that is related to the influence of nitric oxide. One last part that I would like to mention is diet and lifestyle. We eat too much and we eat too frequently. I'll say that right away, in average. And I think that is affecting the way that our body cleanses. And therefore, we have less time available for detoxification as well. And we don't have enough nutrient density. And if you go to the SAT diet, even worse. And we don't put too much fiber in our diet that will definitely feed the healthy microbes that are doing all these fantastic functions in terms of helping detoxifying. So we need to support that. So polyphenol intake, a fiber increase, a micronutrient diversity are some things that we need to consider when we speak about healthy diet, but also time-restricted feeding. Don't skip breakfast, but skip dinner. Eat early. The last meal of your day has to be very early, hopefully 5 p.m., 6 p.m. top. And then you will get a good amount of time without eating through the night where this detoxification activation happens so intensively. And you can eat your breakfast next day because you're going to be hungry, but you had this intermittent fasting time that is fantastic for detoxification purposes and microbiome balance altogether. And lifestyle, well, we're too clean. We need to get a little bit dirty. We need to go outside and get a little bit of this nature inside around our bodies and And that's super important. So nature connection is something that we need to get on a frequent basis. Eliminate anything can have antimicrobials, chlorine, all these things and chemicals. Try to get natural, organic products in terms of house cleaning products and anything related to your bedding and mattresses and pillows. Try to make the change gradually to more cleaner options. I would say get a dog, but that's just because I love those guys so much. And they also, actually, there's research done that they can have a very important effect in your microbiome profile in a positive way. Protect yourself from EMF. It's such a huge deal right now. And try to get wired. And wireless has to be hopefully reduced to the minimum expression, especially in our house. Stress reduction, any form, any type you can get, amazing. Air purifiers, I think they're mandatory. We should have one at least in our bedroom. And we get so much pollution from the environment, and I think we need to purify our air on a consistent basis nowadays, especially if you live in a more dense populated area. It is a reality. I don't like the idea, but I think it's a necessity at this point. Water purifiers, same thing. You can't drink your water out of the tap. It's loaded with chemicals. Go to ewg.org, put your zip code, and see what the water looks like. You will be, unfortunately, dazzled by the amount of toxins that you can find. Of course, organic eating, local, ideally regenerative foods, even better, and get clean, get organic, and protect yourself, inform yourself, because avoidance, again, is the key for detoxification. Well, that was all. If we want to improve microbiome balance, we need to take care of the hypertoxicity problems we're facing nowadays. Thank you so much. Hopefully, talk to you in another opportunity.
Welcome to Rupa Health, the best place to order, manage, and track results from over 30 different lab companies in one single place for free. It's going to take you under 2 minutes to sign up, and you can order any functional medicine lab for your client in under 30 seconds. Let me show you how it's done. So here's our beautiful interface. I'm typing in the name of my client, selecting the lab that I want to order for them, and hitting send. From there, Rupa and their amazing team handle the rest. They email the client, collect payment, and even offer an interest-free 3-month payment plan. We've also built the world's largest library of information about chronic health conditions, the lab tests that can help you find the root cause, and the evidence-based interventions that you can use to help people heal from them. It's called the Rupa Health Magazine. There, we have in-depth articles about almost any health condition you can imagine. And we give you step-by-step protocols that other clinicians have used to help their clients heal and that are verified by evidence-based sources. You should totally check it out, and it'll transform your practice. And we can't wait to see you. So make sure you sign up today at rupahealth.com.